Hello, my name is Nisha Danya, and Group 2 will be talking about seed development with the title A Friend in Seed is a Friend Indeed. So in Group 2 we have 4 people, Nisha Danya, Marlia, Hiroshini, and Nadira. So in the video we'll be talking about stages of unicote development, zygote and pro-embryo stages, globular stage, heart stage and torpedo stage, the mature embryo, and monocot versus unicote. So what is a seed? Well, seed is a reproductive part of a plant. It contains the embryo and stored food surrounded by the seed coat. So when the seed develops, the plant will undergo cell division, cell elongation, and differentiation into tissues and also organs. So in the video, we also included an interview session with Ms. Nickner Ayn Ramley. She's an agronomist for Greenwell Genetics. So we were given the chance to ask her job as an agronomist, her job specifics, the usage of clean seed production concept, the technologies used by Greenwell Genetics, and also the most exciting part of her job as an agronomist.
Alright, so my name is Nurul Madlia and I'm going to describe about the zygote stage and also the pro-embryo stage. So the zygote stage starts after double fertilization. Immediately after double fertilization, the zygote and the endosperm will become visible. So the zygote is very small and it has a dense cytoplasm. So during the zygote stage, the zygote will divide repeatedly in all directions, which will form several cells called pro-embryo. So moving on to the pro-embryo stage. So during pro-embryo stage, there is an elongated structure called the suspensor. The function of the suspensor is to transfer and produce nutrients from endosperm, which will then allow the embryo to grow. So that's all for the zygote stage and also the pro-embryo stage.
So right now we are moving to the globular stage. In this globular stage, we can already see we already have the shoot and root exists. So for the root, it is formed at the embryonic cells near the suspensor, and the other end will become the shoot. The dermal tissue in this globular stage is formed from the outermost cells of plant embryo, and the outermost cells of this plant embryo will divide with cell plate, and hence it will produce the single outer layer of the cells. And the function of dermal tissue is to protect plants from desiccation and also including the stomata. Next, we have heart stage and torpedo stage of embryos. In the heart stage, we can see that the embryo is in a heart shape. This one is in a heart shape. So this happened because of the rapid cell division that occurs in the cotyledons or seed leaf. Next, the torpedo stage. So in this torpedo stage, the embryo is in a torpedo shape, and we can already see the shoot and root apical meristem. This happened because of the enlargement and elongation process that occur in this stage. The shoot apical meristem is for the above ground growths, and the root apical meristem is for the underground growths. So that's all from me. I'll pass to the next presenter.
Okay, a very good day everyone. Today, now I'm going to present about the mature embryo. Okay, first of all, here you can see the structure of the mature embryo. There are several structures that can be found in the inside of the mature embryo, which is first of all plumule, where it can be found at the tip of the epicotyle, and it consists of shoot tip and small leaves. Furthermore, we can also find epicotyle, which contribute to the shoot development, and the hypocotyle contribute to the stem development and terminates in radical or embryonic root. Furthermore, we can also see procambium, which is found at the core of the embryo, which will form vascular tissue that contribute to the water and nutrients transport. Moreover, we can also find seed coat on the embryo, which enclose and protect embryo and its food supply. Then we could see the cotyledon, which will contain stored food reserves of seed. Then now I'm going to talk about the monocot versus eudicot seeds. So as you can see here, this is the eudicot seed and this is the monocotyledon seed. What are the difference between these two is the number of cotyledons, where the monocots have only one cotyledon but the eudicot have two cotyledons. Well, in the aspects of how nutrient molecules are stored in seed is, in monocots, cotyledons store only certain nutrient and absorb other nutrient molecules from endosperm and pass them to embryo. However, in eudicots, the cotyledons store all the nutrient molecules the embryo uses. Well, for the presence of endosperm at maturity, monocots it's still present, while for the eudicots it disappears because it is taken up by the two cotyledons. That's all from me. Thank you.
Good evening. My name is Nisha Danya, and today with me I have my group members, Nurul Marlia, Kiroshini, and Nadira. Shall we begin? All right. My first question is, could you give us a rough picture of your field of expertise?
I'll introduce myself first. My name is Nur Ain Ramli. I am agronomist for Greenworld Genetics Sdn Berhad, which is located in Cameron Highland. So mainly Greenworld Genetics is a company which focus on selling tropical hybrid seed. And we do research and development, take data, the growth performance. Lastly, I will submit the final report.
Yeah, that was very interesting. Thank you. Thank you. So, I read that Greenworld Genetics utilizes the clean seed production concept. Well, what is this concept and how does this concept help to produce better seeds?
Clean seed production concept is meant by reproducing pure seed. The concept slightly affect the seed development. We need time to make sure the inbred that we use is pure, so the seed that we produce is also pure.
My next question. May I know what is the most cutting edge technology that is being used by Greenworld Genetics to produce better seed?
Greenworld Genetics now have biomolecular lab, which we use DNA marker to test on corn seed. We want to know the purity of the sweet corn seed, and then we do it on the field. We mix the method with conventional method and the biomolecular method.
So before we wrap up this interview, could you share with us what makes your job most interesting in your point of view?
I love my job very much because I live in Cameron Highland, which where a certain crop only can grow in good condition here. Then so I think my job is cannot be found anywhere except in geology.
That was very interesting. Thank you so much, Ms. Ayn, for the sharing. I've gained a lot of knowledge through this interview. I think my group members as well.
Yes.
So
Definitely. Thank you.
we really appreciate your time today. Thank you so much.
Yeah.
So in the video we talked about how monocots are different from eudicots. This is because monocots have only one cotyledon. Besides that, we talked on what happens after fertilization. So after fertilization, the zygote will rapidly divide to form proembryos. Furthermore, we talked on the heart stage and the torpedo stage. So in the heart stage, the cells will divide rapidly, while in the torpedo stage, cell elongation happens throughout the embryo. So, in the interview, we talked about GWG, who carries out research and development and also sells tropical hybrid seeds. Besides that, Ms. Nigner-Ein explained to us about the clean seed production concept, which is used to ensure the purity of the seeds. Besides that, she also talked about how Greenwald Genetics uses DNA markers to find out the purity of the seeds. Thank you from Group 2.